Hello, everyone, and welcome to this afternoon seminar. Our speaker is Dr. Rita Brookheart, who is currently an assistant professor at the Division of Pediatrics and Nutritional Sciences at the Department of Medicine, Washington University School of Medicine in St. Louis. Dr. Brookheart received her PhD in molecular cell biology from the same university, where she was awarded an NIH NRSA F31 pre-doctoral fellowship. Completed a postdoc in the lab of Professor Peter Espinshade at Johns Hopkins School of Medicine, where she received an NIH NRSA F32 individual postdoctoral fellowship. As a faculty member of WashU, Dr. Brookheart has been awarded several grants in support of her research, an NIH K-12 award, an NIH NHLBI K01 Career Development Award, and Washington University NORC Pilot and Feasibility Awards. A primary focus of Dr. Brookheart's lab is understanding the intersection of metabolism and stress responses, human health and disease, in basic and translational approaches. In addition to research, Dr. Brookheart enjoys mentoring and encouraging the next generation of scientists. She has 2 patents to her credit and has published in high-impact journals such as Cell Metabolism. Dr. Brookheart has very impressive awards and honors under her belt, such as the 2022 Vanderbilt University Emerging Scholar and Harvard Medical School NORC Diversity Scholar from 2021 to 2020. She served as an ad hoc reviewer for the SMEP study session, and also early career reviewer for the POMD study section in 2022. Has served as co-organizer for the NIDDK Step Up Symposium since last year. Today, we are honored to have her here to give a seminar on the topic, molecular players linking. Okay, welcome.
Thank you so much for that introduction. It's very nice. And thank you for inviting me. I've been having a really wonderful time talking to everyone and really getting immersed in exercise and nutritional science. It's really nice to have all these chats. I've learned a lot and it's been really great. So today I'm going to talk about muscle and particularly looking at metabolism and control of muscle mass. So we're all pretty much well aware of the importance of mitochondria to controlling the body's response to stress. And obviously there are lots of different stressors that we come in contact with. Obviously physiologic stress that we encounter during development, organismal development, obviously exercise, which everyone is aware of here, as well as natural aging processes. However, there's also pathologic stress, right? So with regards to heart failure, where the utilization of fuel in the heart can change, right, to a more glycolytic stance instead of a more oxidative stance with fatty acids. And as well as the alterations in cancer that are appearing, especially with regards to hypoxia, where the decreasing availability of oxygen can alter the composition and synthesis of various types of lipids and sterols. And obviously, with regards to muscular dystrophy, this obviously places a significant amount of stress. So it's pretty much one of the main jobs of the mitochondria is to really respond and adapt to these changes in cellular demands. And all of these stressors can elicit various demands. Just highlighting a couple here with regards to change the amount of energy that's required for the body or for specific organ systems can also impact the mitochondria response. Changes in the availability and the types of nutrients that are available to these cellular structures, such as changes in the availability of specific amino acids that are present and usable as a fuel source can vary with regards to different stressors. And one of my favorites, obviously, changes in the availability of lipid species. So the types of lipids that are available can actually change the membrane fluidity of various organelles, particularly mitochondria. And we all know that could have detrimental impacts depending on the types of lipids that are incorporated. So as I mentioned, all of these changes that different types of stressors, physiologic, pathologic stressors ensue, can likewise create stress to the mitochondria. And this can obviously significantly impact both the function and metabolism of these mitochondria in response to these stressors. And several studies, much work has been focused on defining the pathways by which physiologic stress, I forgot there's a microphone, physiologic stress as well as pathologic stressors impact mitochondrial function and metabolism. So there's lots and lots of work that we're all really well aware of, and lots of us do contribute to this beautiful work. However, there's a lot that we don't yet know, right? So there are several unknowns that remain as to how do these stressors alter these mitochondria, and what is the endpoint of this? And so that's kind of a phrase that a lot of us use in science, right? Like these functions remain unknown. This pathway remains unknown. This synthesis remains unknown. And so with all of these unknowns floating around, we might feel like Captain Kirk here, where we're just kind of aimlessly going through this galaxy of unknowns, and not really seeing clearly, is there an end to this? You know, like you may answer one unknown during your doctoral work, postdoctoral work, even as a faculty member. Hopefully you answer more than one, but you answer one unknown and you feel great. Like I figured this out. I know what's happening in the system. This is just, it's such a prideful moment for us. But then that known leads to even more unknowns, more questions, right? Well, it kind of seems endless. But I mean, that's why we all do science, right? To figure out these unknowns and to sort them out and to answer more and to lead to more unknowns that we hope to answer, but maybe the generation before us, the scientists after us will then answer, hopefully, and we'll build upon our own work. I mean, that's how science works. That's why we come in every day. So today I'm going to start off with, I keep going back and forth, sorry. Today I'm going to start off with an unknown in line with this theme. However, it's an unknown that's a little more personal, a little more tangible, right? And that's because it's an unknown that started with a patient. So an actual person, not just cells in a dish, not just a rodent model, a pig model, right? Like it's an actual person that we can talk to and know. And so this patient presented to our ER with extreme muscle pain. And her history is rather interesting because she reported to the ER when she was 24. And at the time, she had a very lean BMI. And prior to this occurrence, she was very athletic, both in high school and in college. She would do track and field, volleyball, any sport, you name it, she did it. And she was great at it. However, once she got older in her 20s, she hit a wall. And so after college, she became exercise intolerant. She could no longer run. She could only tolerate low impact yoga, which if you saw where she was coming from, that's just such a huge decline. And this is still in her 20s. So she's a very young woman at this point. She had persistent muscle weakness that was exacerbated by any form of extreme exercise. And she had frequent trips to our ER, complaining of muscle weakness and fatigue. She couldn't fast because then she would experience high degree of fatigue. So it was a really just unfortunate occurrence. And so one of the first things that our pathologist thought was, well, let's look at her muscle, right? Because exercise, you think the cardiovascular system, you think of muscle initially. So they were able to actually obtain biopsies from our patient. And obviously H&E staining looked normal. Fiber typing looked absolutely normal. Lipid depositions looked normal. Various mitochondrial readouts for mitochondrial activity looked normal. However, what our pathologist then did was, because he loves EM, he thought, let's put this under the EM and take a closer look. And one of the things he did notice were these giant structures, these globules, kind of sporadically placed throughout the biopsy. And we weren't really sure what these were. And his best guess was, okay, maybe these are some sort of lipid species, some sort of like lipid related composition is happening here. And so then he took a closer look and looked at her mitochondria. And what he noticed was these mitochondria really had kind of weird shapes and kind of irregular shapes. And so this kind of led the clinicians into thinking, okay, well, this is probably a mitochondrial disorder, right? Because that would explain the lack of exercise, yada, yada. And so they ran all of these tests. They analyzed mitochondrial function. And bear in mind, this is in patient biopsies. Mitochondrial function, just looking at sort of the enzymatic activities, everything looked fine. Everything came out fine. So they thought, okay, well, to date, it is not a known mitochondrial disorder that she has. And we still don't know what's going on. So we decided to sequence her genome. And so as a control, we sequenced both of her parents. And we had two geneticists, a pediatric geneticist, as well as geneticists for adults doing these studies. And one thing that they noticed was this mutation here in this particular chromosome. And they were able to show that this particular mutation, this particular allele change was actually in the gene that encodes for site 1 protease. And so we thought, okay, well, we noticed this genomic mutation. Are we actually observing, just because you have a mutation doesn't mean you're actually going to generate RNA from it, right? Maybe the RNA gets decayed, maybe it's expressed at a different rate. So we got isolated her RNA and obviously synthesized into cDNA, and then we did some more sequencing, and we were still able to see this mutation. And our geneticist did some bioinformatics, and he was able to show that this actually occurred at roughly a 50% frequency. So it appeared that indeed this, obviously it's a heterozygous mutation, but the actual expression of this mRNA off of this mutated gene is occurring at a 50%. So indeed it is a heterozygous mutation that's then producing 50% of the RNA as a mutated transcript. So this was interesting, but I mean, just because you're making RNA doesn't mean it's doing anything, right? It could be getting, I don't know, it could just not be functional. So to do our sorry, I don't have the prompts of where I'm going. So I'm kind of like, so bear with me. So before I get that, I'm getting ahead of myself. What is Site1 protease? Some of us know. I found out a lot of people don't know outside of my bubble. And so I'll just cover that here. If you guys do know, just bear with me. So Site1 protease is known to control development and human disease. We think it's pretty important in development because if you try and make a whole body knockout, you don't get any pups in mice. And so to date, no one's really sure what's it doing in development. That's something that my lab's actually focusing on with regards to S1P, regarding myogenesis, et cetera. It's been heavily studied in the liver and bone. There's beautiful work from UT Southwestern, from Brown and Goldstein lab, that's really characterized S1P, as well as Jay Horton. It's a group at UT Southwestern. What's been known is that it's required for cartilage formation during development in mice. There are several mutations associated with S1P that have been shown to lead to skeletal dysplasia in humans. So there's a human link to disease as well. And recent work has shown that cleavage or activation of S1P is elevated in patients with NASH or NAFLD. And most recently by our group, which we published this a little while back, we were able to show that a mutation in S1P is actually associated with a disruption in exercise tolerance. And that was the work that I just kind of briefly touched on about our patient. So I'm going to kind of take a little closer view of exactly what is S1P doing in the cells. We know it has an important role in human disease, and it's really heavily studied in these two kind of organ systems, skeleton, as well as, the liver. And so just kind of like to dive down deeper, I love signaling pathways. This is just kind of an overview of what is S1P doing in the cell. So S1P, which stands for Site1 protease, is actually important for the regulated activation of several transcription factors crucial to cellular pathways. And so I've highlighted some of those here. Site1 protease, which is beautifully shown by Brown and Goldstein's lab, cleaves and subsequently activates the SRBPs, which are required for sterol and lipid uptake and biosynthesis. Site1 protease is also required to activate one of the three arms of the ER stress pathway. It activates ATF6. It activates several of the CREV family proteins to do all of these wonderful things I won't go into because it's not really related to the talk today. And it's also required for lysosomal biogenesis. This is not a transcription factor. It's rather a subunit that then gets trafficked to the lysosome. The only way it can get trafficked to the lysosome is if F1P cleaves it and liberates it. So S1P does lots of different things to lots of different cell types in lots of different ways. And it's a very important protein, I think. And I think lots of people do too. Now, how is it functioning? How is it activating these SRBPs, these ATF6s, and these CREB proteins? So S1P actually exists in the ER. Some people have called it like a pro-S1P, so it's not yet cleaved. And so once it's in the ER, it undergoes a series of autocatalytic cleavage events, which Brown and Goldstein have touched on in previous papers. These autocatalytic cleavage events then allow S1P to travel to the ER, where it is now seen as being activated. Exactly precisely the mechanism by which S1P is activated is still being figured out. But in the Golgi, it's seen as an active component. While it's in the Golgi, it is now in proximal location and it's active to various transcription factors, such as the SRBPs and the ATF6s, which I should mention, those also can translocate to the Golgi under various stress conditions or the availability of different sterols or oxysterols. So you have all these components here in the Golgi. S1P is active. It can then cleave it can then cleave either SRBP or the ATF6. And then what then happens is another protease, Cy2 protease, can then lead to a second cleavage event for both of these transcription factors. This then leads to the liberation of these transcription factors from the Golgi, so they're no longer tethered to the Golgi, and they can then translocate to the nucleus, where they can then bind to their favorite promoters and activate all of their favorite genes. So as I mentioned before, Site1 protease is classically studied in the liver and in the bone. And so in looking at our patient, we asked, well, is S1P doing other things in other places in the body, particularly in the skeletal muscle? And the reason why we really asked this question is because we have this patient, they're exercise intolerant, they have myoedema, they have elevated CK levels that are just through the roof, and they have these really enlarged SM mitochondria. So it was really pointing us in this direction. So we decided to explore this further. And so this is just showing you the protein sequence kind of focused on the region of the mutation for our patient. And this denotes the transmembrane domain region for S1P because it's a transmembrane domain protein, forgot to mention that. That's how it can tether itself to the ER and then move to the Golgi and tether itself to the Golgi. And what our patient's mutation encodes is amino acid switch from a proline to a serine within the transmembrane domain region of S1P. And proline is generally thought of to be a helix breaker. And so the transition from proline to serine is predicted through in silico to be pretty detrimental to the function of this protein. So one of the first questions is this mutated S1P doing anything? Is it functional? Is it non-functional? What's happening? So the first thing we did is we wanted to see, can we even express this mutated mRNA sequence? And so what we did is we were able to acquire these cells, these SRD12B cells from Brown and Goldstein at UT Southwestern. These are a Chinese hamster ovary cell that they made a while ago. I didn't put the year on, but it was a while ago. And I'd read about it all through grad school and I was really excited to use these cells. But so we got these cells, and what they did was they actually made these cells so that they don't express S1P protein. And so we thought that would be a perfect kind of blank slate to test, okay, is this mutated S1P protein expressed at all? And so what I'm going to show you now is just a Western blot, just showing this is staining for probing for S1P. This is our control, so just empty vector. This is just a non-specific band for our antibody. We aren't able to visualize any S1P in these cells, which Brown and Goldstein published that. However, when we overexpress our mutated protein, we see it. It shows up. And obviously, as a positive control, our wild type S1P also shows up. So that was like a relief. And then alpha tubulin is just a loading control to make sure that everything's loaded the same. And then these characters, A, B, and C, are just the different kind of forms of S1P as it's being cleaved, as I mentioned, as it travels through the cell. And so we were actually able to see these different forms in our mutant protein, as well as obviously in the wild type. So this mutated gene is producing mRNA at roughly 50-50%, right, representation, and you're getting protein off of it. So the next question is, just because the protein's there doesn't mean it's doing something or what it should do. So we first started off by looking at the localization of this protein. So as I mentioned before, S1P localizes to the ER as well as the Golgi. So we did some IF. This is using cells that are just expressing like wild type S1P or our mutated patient S1P. And what we were able to show is, again, S1P is here in green for both of these. The top panel, we're also co-staining with a KDEL, which is a marker for ER. Down here is GM130, which is a general marker for the ER. I believe it's a trans-Golgi network. I don't remember, but it's a part of the Golgi. And so what I think you can appreciate is that we are able to see, as has been published by several people, that S1P is able to co-localize with the ER, wild type, as well as with the Golgi. And then when we look at our mutated S1P, we do see some co-localization, but it appears to kind of be all over the place. And we do also see localization in the Golgi. And so presently we're trying to figure out, okay, where are these other places? There was a paper that came out, I believe it's last year or so, that was showing that at least in human keratinocytes, S1P can localize to the ER. So we're also kind of trying to investigate that. Do we see that in our system, or is it specific to these? There were immortalized keratinocytes. So we were trying to see if that's like a specific thing that they observed, or if it's translational to other systems. So the protein's expressed, it goes to the Golgi, so presumably it's active. But we really wanted to test this, right? Because just because the protein's where it should be doesn't mean it's doing what it should. So as I mentioned before, S3P pathway, ATF6 pathway, if S1P, mutant S1P is in the Golgi, we wanted to then test, okay, well, is it activating SREBP? Are we seeing an increase in these lipid and sterile biosynthesis genes? Are we seeing an increase in lipid and sterile biosynthesis? So that was kind of our readout of one of the readouts we use for functionality of this mutant. And this is a paper by Brown and Goldstein, where they first isolated these SRD12B cells that I mentioned. And they actually did this really beautiful assay using crystal violet. And so crystal violet is just this dye. You have cells on a plate. You put this crystal violet, it's a violet dye on the plate, and it'll stain like all the cells on the plate, all the cells that are presumably alive and adherent to your plate. And so what they did was, is they had these CHO cells, Chinese hamster ovary cells. This is the parental strain, so like quote unquote, they're wild type. Then they were also able to generate another, from this line, a CHO cell line that is deficient for S2P, and then two other lines that are deficient for S1P. And they noticed that when you have full media, so you have cholesterol in the media, mevalonate, which is supposed to trigger the S3P pathway, and oleate, so fat, in the media, everyone is happy. You see purple, well, black, right? Because this is a while ago. You see black growing, which is crystal violet. They're crystal violet positive. So presumably these cells are attached and they're happy. When you take away all of these additives and you just have pretty much, F12, because that's what these guys grow in, with DMEM, as well as LPDS, so media that does not have any lipoproteins in it, right? So no fat, no cholesterol. The wild type cells grow happy. S2P is gone. These cells are gone. They are off the plate. The two S1P mutants that lack S1P are also gone, presumably because they cannot make sterols. They can't make their own sterols. They're sterile oxytrophs, meaning you have to artificially provide them sterols and fats to use because they can't produce it on their own. So we thought this was a really cool experiment. I think you can tell because I keep talking about it. So I wanted to recapitulate this, but in our mutant, to see, okay, does our mutant grow when we take away these sterols, when we take away these lipids? And so we kind of repeated the same experiment. Here we have our control, or like empty vector transfected cells. These are the same cells that we got from Brown and Goldstein. Here we have, it only says sterols here, but it's the same thing. It's mevalonate, it's oleate, and it's cholesterol. The cells grow happy, right? Sorry. Okay, gotcha. The cells grow happily. I need my, yeah. To print them out and be like, okay, Rita. Sorry. So still, you add the sterols and fats, they're very happy. You take away the sterols and fats, so it's just DMEM, F12, LPDS, they're not happy. These cells are just floating off the plate. There are no cells on the plate, evidenced by the fact that there are no cells to stain purple. We add the wild type S1P back. Just like Brown and Goldstein, we were able to like rescue this phenotype. Then we asked, okay, what's going to happen when we add back our mutant S1P to the system? They also grow. Very upset about this. And we repeated it like a dozen times. I said, no, did you mix the cells up? Did you add the wrong, did you add wild type to this? And then I did it, and then two other people did it, and we got the same result. But I mean, that's why we do science, right? You want to make sure what you're getting is real. And so it's real. So these cells were able to rescue the sterile and lipid phenotype by overexpressing this mutated protein. And this kind of gave me a pause, because usually when you talk about mutations in patients, mutations in mice, you always think of a loss of function, right? We are inhibiting this protein. This is why we see a phenotype. Here, this is suggesting maybe the protein's functioning, but functioning in a different way, and that's causing the phenotype. So we decided to drill down a little bit more into this SRP pathway. I like SRBP a lot. That's why I spent so much time on this. And we decided to look at this transcriptional readouts here to really get just a little more understanding of what's happening. And so what we did is, again, Right. So here we're using fibroblasts we collected from a control patient as well as our patient. And we were able to validate that, yes, the mutation's there, everything's there. And so here we're treating these cells either with zero mevalonate or with increasing concentrations of mevalonate. And as I mentioned before, this is to stimulate the S3P pathway. So try and stimulate S3P moving to the Golgi to get cleaved by S1P and turn on all these wonderful genes. And this is just qPCR looking at different genes. So there's ACC. Right. There's farnesyl diphosphate synthase, there's HMG-CoA reductase, squalene synthase. So all these different components as the different S3Ps are supposed to be turning on when you trigger the pathway. What I think you can appreciate is that in our control cells, we kind of see like some increase, right? It's increasing when you start trying to trigger the pathway for S3BP. But in our patient cells, even at baseline, for several of these readouts of S3P activity, there's more transcript being produced, or there's the abundance of the transcript is increased rather, right? I'm getting ahead of myself. And so we thought this was really interesting. And then when we try and stimulate the pathway, like full on, push the gas, we still kind of see the same trend where we're producing, or there's more abundance of these S3BP transcripts in the cell. And so that was really interesting. So pretty much what we concluded was that this mutated S1P is somehow having enhanced S3P transcriptional activity. And we decided to do just a quick luciferase assay reporter using fatty acid synthase, luciferase reporter, which would get activated by S3BP. And even at baseline, we see the similar trend. So just in cells sitting on the plate, we see increased luciferase activity, which would suggest that you're seeing an increase in S3P transcriptional activity. So that was pretty interesting. So we thought maybe this is a gain of function mutation, and maybe this gain of function, this overabundance of S1P activity is somehow impacting our patient. But as I mentioned, S3P isn't the only gene that S1P activates, right? It also activates ATF6. ATF6 is required for full-on activation of the UPR, or the unfolded protein response, in response to ER stress. So we decided to do the same experiment as with the mevalonate, but this time instead of adding mevalonate, we induced ER stress by adding tunicamycin to these cells, and we did qPCR, and we looked at GRIP78, this should just be XBP, and CHOP. So these are genes that are traditionally activated by ATF6, as well as sometimes other arms of the pathways. And what we saw was, we kind of saw the similar trend where there is an overabundance of these transcripts in response to ER stress. I want to take a side note. So if you remember, before we added mevalonate, just at baseline, we saw an increase in S3BP transcripts. Before we added tunicamycin, we did not see an over increase in any of these ER stress response genes. And we thought that was really interesting. Like one pathway is active, the other one's not, just kind of at baseline. And so we're still trying to figure that out. Like, what about this mutant is able to presumably selectively activate certain arms of these transcription factors under basal conditions? So when no one's stressed out, there's no need for more lipids, there's no need for ER stress to respond to regain proteostasis. So this is kind of our working model of what we think is happening and kind of where we're trying to go from here with this patient. So as I mentioned before, S1P gets liberated, goes to the Golgi, and can activate all of its favorite genes. What we think is happening is that this gain of function mutation, maybe there's more of it localized to the Golgi. Again, we are hypothesizing, right? Because I didn't show you any quantification of this, but that's kind of where we are working from right now. I also mentioned that S1P has been shown by another group that it can also presumably localize to the mitochondria. So that's also something we're looking at. Maybe localization is somehow driving this response that we're seeing, but we aren't really sure yet. We don't know. And so we thought this was really interesting. Yes. Mm-hmm.
So getting more and more material.
We've done both. So we have isolated human fibroblasts from the patient and from a control patient. And so we're able to culture those to a limit. So we only have a certain number of vials frozen down. because I think we can only split them. I don't remember how much, but I mean, it's limited. It's less than like 5 or 7, and then they're done. We don't want to use them anymore. And so we also have cloned this into our CHO cells that don't have S1P. So we're kind of using both systems. Just for skin fibroblasts. So we just did a skin puncture. Yeah, I would love to have gotten enough from a biopsy to get muscle, right? To start growing the satellite cells and to try and differentiate those. But we were only allotted the one punch from the biopsy to do the histology. So. Yeah, so I kind of forgot that pathway. I used to know it perfectly because we used to use mevalonate all the time. So, and help me if I'm forgetting, right? Because it's like, I'm trying to draw it out in my head. So pretty much, I think, so when you have your starting material, mevalonate, you can switch over into 2 pathways, right? One that's less abundant because the cell needs mevalonate, right? Otherwise it dies, right? So you have to have that anyway. And so if you're adding in that mevalonate, I think the cell senses there's too much mevalonate and you don't have enough sterols, and so somehow it activates SRBP.
Those both hormones and cholesterol. That split, so it's a powerful indicator of flux. Yeah, like you said.
Okay. I used to know exactly the pathway, and I could draw it out for you, but I have not thought it out.
37 reactions in there.
It's a bit much. So yeah. So classically, people use that to sort of jumpstart and trigger the activation of SRBPs to make more cholesterol to account for the fact that you're shuttling the mevalonic. It's kind of like a lipid species, yeah.
Got a lot of carboxyls on it. It's acetyl-CoA plus acetyl-CoA. When you're making cholesterol, it's pretty high at the top.
Yeah, it's before you get, yeah, nothing. Thank you for helping, because I have not looked at that in a long time. Right, and so we have this cool patient phenotype, and we're still trying to explore this. We want to actually make a CRISPR knockout that mimics the mutation to really figure out, well, what's happening to this patient, because we can only do so much in our patient, obviously. And so this still doesn't answer the question of what is S1P doing to skeletal muscle. And so to get to that, since we only had one patient, we decided to make a mouse. And so in this sense, we made a knockout mouse. And so to do this, we used an established S1P floxed mouse that Jay Horton at UT Southwestern had generated. And fortunately enough, another PI at WashU had these mice. And so we were able to ask Jay Horton, hey, can we use the mice that's across the street? Can we just get the mice from across the street instead of you trying to send us mice or us trying to go to Jack's and they're cryo preserved and it'd take forever? He said, sure. And then that PI just gave us the floxed mice. So that was really nice and generous. And then we were able to get these HSA Cree mice from Jackson Lab pretty easily. And so we were able to cross these mice together and we were able to generate our skeletal muscle specific S1P knockout. I should mention HSA Cree, it's human skeletal muscle actin Cree recombinase. So it will only become expressed in the skeletal muscle. So in myofibers. So it won't hit satellite cells or FAPs or anything, like it's specific to the myofibers. So one of the first things we did is we kind of just did a qPCR to look at the expression of S1P in these mice. And we looked at gastrocnemius, soleus, liver, heart, and kidney. And what I think we can appreciate is the expression of S1P is significantly decreased in both of these muscle types. However, we don't see any impact. Thankfully, we don't see anything on the heart because that was an issue with other Crees or any of the other organs, especially the liver. And so, as I mentioned before, S1P is important to activate SRBP, and SRBP activates sterol and lipid biosynthesis. So this is kind of where we started to characterize these mice. So we looked at their body weights, we looked at their lean mass, fat mass using echo MRI, and we saw no change. These mice look indistinguishable from their litter mates, their wild type litter mates. We looked at plasma, tag, and cholesterol levels. Everything just looked unremarkable. Everything looked normal. These mice seemed normal, activity seemed normal. We then decided to look at different, transcript levels of ATF6, as well as transcript levels of the SRBP pathway. And this is in the gastrocnemius. everything looked normal. There was really nothing that jumped out at us. So then we decided to take a closer look at the actual muscle. And one of the first things we did is we actually weighed the muscles. We weighed, these are male mice, I should mention, 12 weeks of age. We measured the soleus, gastrocs, and the TAs. And we didn't really notice any difference, except it appeared that the gastrocs were heavier. So we thought that was interesting, kind of weird. Okay. So we decided to do some H&E staining of the gastroc. And what I think you can appreciate is that this is just the wild type littermate, which is the flox mouse gastroc. And then this is the knockout gastroc. And what I think you can appreciate is there are these centralized nuclei that I pointed with the arrows. And they're kind of just littered throughout the gastroc of these mice. And right now we're actually trying to do some regeneration studies, injury models, to actually see, well, is this beneficial? What's happening? Is there some sort of weird response to muscle injury that these mice might be susceptible to? But that's still a pretty junior story. But yeah, I just wanted to mention that we thought that was really interesting that we see these centralized nuclei. And we aren't really sure, is this good or bad? We don't know.
So, as you know, centralized nuclei are all.
Yes,
But
right. We didn't see any.
so
We could be. Yeah, that's a good point. Yeah, we don't know. You mean like us artifactually doing something, or?
consequence of your knockout and lesion that
It must be. Yeah, it could be doing that. Yeah.
the fibers don't look damaged. At least
No,
in
they
those,
look great.
but, you know, muscle
Right,
regeneration
is right,
that's
the central.
what it's used for.
Right, exactly. Yeah. And so we're actually doing some studies to do various injury models. We also have a PAX7 Cree that we crossed our flox mice to see if the satellite cells are impaired. I know it doesn't get at this question, but it's kind of led us into looking at regeneration.
I gotta ask.
Mhm.
These animals actually-
The exercise, great. I didn't include that data. I should have. I know everyone was like, you should include the exercise data, Rita. But so these mice exercise like crazy. And so it's kind of the which doesn't, right. It doesn't make any sense. I know. But we thought it was really interesting because
So is this the knockout? So this is not the defective S1P.
no, right. It's the complete knockout. Like
This is the
Right.
complete, which that shouldn't then be surprising, because if the defective S1P is doing something to inhibit exercise,
Right.
you may have taken it away.
right. And so now they can exercise more. So
Does this have to do with your patent?
the first one, maybe, yes.
Yeah. Very cool.
And so, right. And so, the patient had presumably, lost my place. Sorry. Yes. No, it's good. It's good. It's fun. I like the interruptions. It's fun. It's like lab meeting. I like it. I like lab meeting. It's like, Rita, what's this? Right. So she had a gain of function of S1P and she couldn't exercise. And so the cool thing about this is that we're having a loss of function, right, of S1P, and these mice just exercise. And so this is all male data, but we also have female data that recapitulates this, actually even more so because their muscles are actually even heavier compared to the males. And they just run. And the female mice run even more so than the male knockouts. So we're still trying to figure out, we're not really we think it's cool, but we need to get collaborators and figure out what's happening, because we don't really do looking at males versus females sort of stuff in the lab yet. So, but anyway, right. So we're not really sure what's happening, but we are trying to explore it. But we thought this was a cool phenotype, and we said, okay, this is interesting. And so COVID hit, and we had all these mice, and they were getting older. And I was like, we can't get rid of all of them because I just couldn't. And yeah, so we had some really old mice. And I thought, okay, well, let's see what they look like. Like, what does the muscle look like, right? We have these mice. There's no point in stacking them and not using them. And so this is just the data I just showed you in the male mice at 12 weeks. These are 2-year-old mice, male mice. And I know I know, and you can't touch them because then they start shaking. It's nerve-wracking. But what we noticed is that the soleus as well as the gastroc in the male mice were heavier. Whereas we didn't see this down here at 12 weeks, right? Because the muscles are still growing at 12 weeks, at least in mice. So we saw this. And the cool thing though, when we look at female mice at 2 years, because we did both, the soleus are heavier. The gastroc are heavier in the females, and the TAs are heavier now, too, in the females. So we're still trying to figure that out. Unfortunately, we couldn't really run these mice because I don't know. For older mice, you have to be very gentle and careful with them, and we're just So anyway, so we didn't run these mice, but we want to at some point. Obviously not these, but And so when we first got these data, it was right after COVID, and I thought, okay, well, I see it as like an N of 1, right? Even though we have multiple cages, but they were kind of all started at the same time, and it's a 2-year study. So what we did is we did a second 2-year study. I know, it took a long time. And I said, okay, it's been 2 years for this second cohort, I guess you can call it, right? Multiple cages. Let's see what happens. And it was a blinded study, like no one knew what the genotypes were. And we saw the same data. So I thought, okay, this has to be real, and males and females as well. And I thought, okay, so this is a legitimate thing, because over the course of 4 years, 2 different cohorts, we'll see it. So we're pretty excited, and we want to explore this further. I think the body weights are about the same, because I think we normalized to tibial length and everything because we were worried about that. But if I remember correctly, I think there was nothing interesting that popped out about body weight. We didn't put them in the echo because I was worried they were going to expire before we could do the experiments. And I was like, let's just do the bare minimum with them before something happens. Because we did have a few that would just not make it in the cage. Right. So right now we're kind of wondering, maybe this is implicated in aging. We don't know. It's an interesting thing to look at. But getting back to the 12-week-old mice, we decided to look a little bit more at the muscle. And so we did muscle fiber typing to kind of this is all gastroc data, to understand why are they heavier? Is it hypertrophy? Like what's going on? Fiber types are different. We don't know. And so this is just showing the different fiber types in the gastrocs. So we did cross-sectional area to see if maybe the fibers were larger. We didn't see any real difference. And then so we also looked at maybe the percentage or the representation of the different fibers is different. Nothing, like everything looked the same. Nothing really stuck out. It was really frustrating because we wanted to understand what's happening. And so we like mitochondria. So we thought, let's put them in the Ouroboros. Let's see what's going on. And so the gastroc is actually composed of oxidative and glycolytic fibers. It's not just one kind of fiber type, or like type 1 or type 2. Like it's actually kind of a mix of them. And so there have been a few reports showing that the basal rate of respiration in the the oxidative, which I call the red gastroc, versus the glycolytic, which is the white gastroc, because literally it's opaque, it's white, and then the red is red. So it's pretty easy to dissect it out on the muscle. The basal rates of oxygen consumption are pretty much very different. So you actually you'll see it in a minute. I think you can appreciate if you start down here, you're starting off right around 50. Here, you're not even hitting 50 in the white gastroc. And so what I think you can appreciate is we looked at the red gastroc and we spiked in pyruvate, and we just ran them through the Ouroboros and spiked in ADP, succinate, and then measured ETS. And we don't see any difference, right, in the red gastroc of these mice. However, when we look at the white, that's where we start to see a difference. And based on these data, and we also added palmitoyl-carnitine as another fuel source to add, okay, is fatty acid, is that different as well? But for the purpose of this, will just show you the pyruvate data. We do see an increase in the respiration of these glycolytic fibers, which we thought was interesting. And so we started to kind of dissect out these fibers and figure out, okay, what's going on with the mitochondria? And so we looked at mitochondrial DNA levels. We also had some other measurements to look at like mitochondrial content. Those were all negative. Like everything was the same as this wild type mouse. We looked at ETC, obviously select ETC components, and everything was the same. Nothing was different, but we have this crazy Ouroboros data. saying that something should be different. So the big question here is, how is S1P controlling muscle mass and controlling metabolism in these glycolytic fibers? So we did some RNA sequencing. And so we got the gastric of these mice, we sent them over to our core, and they came out with 80 significantly differentially expressed genes. And I was surprised there were only 80 and happy that there were only 80. That's like a manageable number. And so we decided to kind of just take a look at these genes and see what was there. One of the genes that really stuck out is MSS51. The expression level of MSS51 based on our RNA-seq data of the gastroc was incredibly low or significantly low. We decided to validate this using gastroc and doing qPCR. And we were able to recapitulate this, that yes, indeed, MSS51 transcript levels are lower in these knockout muscles. What is MSS51? That was my big question. So I sent one of my students out and I said, can you just do like a lit search and like give a presentation and talk to us about what this is? So this is what she came up with. She had lots of other papers, and there's like a yeast homolog, etc. But anyway, MSS51 localizes to the mitochondria. That was interesting. It's induced by TGF beta ligands. Okay, that's interesting. And it's predominantly in glycolytic fibers. What does it do there? It inhibits mitochondrial respiration. So we thought, this is checking all the boxes, right? Like we see an impact on the mitochondria in our S1P knockouts. It's in the glycolytic fibers where we see this occurrence happening, right? In the white gastrocs. And this occurrence is affecting mitochondrial respiration. So we thought, okay, well, could S1P be influencing respiration through this pathway? And so one of the other things we wanted to do is we kind of moved this over into a cell culture model and able to manipulate expression of S1P and MSS51 more easily. And so for this, we use C2C12 myotubes. So C2C12 cells are myoblasts that you can then differentiate into these beautiful myotubes. They're beautiful. I should have brought a picture. They're really cool. And so what we did here was we were able to transfect these cells with either control or scrambled siRNAs, which would not target anything, right? It's our control. Then we transfected in siRNAs, 2 different types of siRNAs that are supposed to target S1P. This says MBTPS1. This is for our paper. The gene name for S1P is MBTPS1, but like the actual protein, et cetera, is S1P. Anyway, so we were able to get beautiful knockdown of siRNA 1 and 2. So we went with siRNA 1 and we looked at MSS51 mRNA levels, and that was also down. So this is able to recapitulate our RNA-seq data as well as our qPCR data on the gastrocs. So we were pretty happy with this. And so next, the big question was, do we see anything happening with respiration? And so for this, we use Seahorse. And again, these are differentiated myotubes that either express or scrambled siRNA, S1P targeting siRNA, so decreasing S1P levels. And then we also threw this in where we decreased S1P and then we spiked in MSS51 using a plasmid. And this is kind of busy, but what I think you can appreciate is that at baseline, here are just, presumably our control cells. When we delete or deplete the cells of S1P, we see respiration go up. And then when we reintroduce MSS51, it goes back down. And then we also see the same trend, the same occurrence in maximal respiration. I can't see that far. As well as in various other, in the other components that we also looked at. So we were pretty excited about this. Because it recapitulated what we saw in vivo, right, in our mice. So now we were trying to get at kind of the mechanism just a little bit, right? Because they're always going to ask about mechanism, and I like mechanism, so we went ahead and tried to figure it out. So as I mentioned before, this is a paper from 2015 from someone else. They were able to show that MSS51 mRNA expression can actually be increased by TGF beta 1 signaling. So myostatin, we're all fans of myostatin, right? We all know what myostatin is. So yeah, you spike in myostatin. These are in C2C12 myotubes, so the same cells that we were using before. They spike that in. MSS51 expression goes up. They add in TGF beta 1, that goes even higher. So we wanted to see, is this how S1P might be modulating MSS51? This was like our working model. Because so far, no one knows how do these ligands activate MSS51. So maybe this is happening. And again, no one knows how this is happening either. So one of the first things we did is, again, we're using these C2, C12 myotubes, where we either knock out scrambled, right? That's our control, or we're getting rid of S1P. And we're doing this either in the absence of TGF-beta1 or in the presence of TGF-beta1. And when I say absence, that's just the vehicle that we use for TGF-beta. And what I think you can appreciate is that when we spike in TGF-beta1, we saw the same thing that the other group did, which MSS51 expression goes up. And we see that in both cases, though. So it goes up when we have our just control cells and add TGF-beta. It also goes up in our siRNA knockdown cells. However, the degree to which it goes up is not the same, right? So it's somewhat blunted. And we were trying to look at this and trying to figure out, are there better ways to knock down siRNAs in these myotubes? And so right now we're trying to work on AAV, because that'll create more of that'll increase our transfection efficiency or infection efficiency, knockdown efficiency, et cetera. So you have the TGF-beta ligand binding to its receptors, and that then leads to phosphorylation / activation of these proteins known as SMAD. These are transcription factors, and they can then go into the nucleus and bind to their favorite promoters. So we decided to ask, is this TGF-beta signaling intact when you take away S1P? We were trying to figure out the mechanism for all of this, or some of the mechanism for it, right? Because mechanism takes a long time. So here we have the same cell situation, right? Where this is just a Western without TGF-beta, with TGF-beta. These are 3 different replicates, 2 replicates, and then 3 over here. And then when we get rid of S1P, we'll deplete S1P, what happens? This is just looking at phosphorylated SMAD2, which is the active SMAD2. It's still there. Like, it still gets phosphorylated even, yeah. Oh, thank you, sorry. It still gets activated even when there's no some S1P. An other arm of the TGF-beta signaling pathway that's known to regulate muscle mass is ATF6 phosphorylation, right? You phosphorylate ATF6, it becomes active. We still see phosphorylation. Maybe it's higher. We have to repeat these more. This is not the best phospho antibody. So we're still trying to figure that out. So based on all this, this is kind of our working model, what we think is happening. We think that you disrupt S1P function in skeletal muscle, you're impacting mitochondrial metabolism, where you see a loss of S1P, decreases MSS51 expression, and this then allows for an increase in respiration. We also think that S1P can be modulating organ size regulation. You get rid of S1P, you see bigger muscles happening. Is there any kind of crosstalk between these 2 streams? We don't know. That's what we're looking at. And the key here would be TGF-beta signaling, presumably. And also, how is S1P changing mitochondrial metabolism and cell size regulation? Like, mechanistically, how is that happening? We have some ideas that we're working on to figure that out. Right, so my lab's pretty much focusing on this, as well as looking at all the other genes that we pulled out of this RNA sequencing data. And I won't go into them, but this is highlighted because right now we're putting together a paper to actually look at S1P's role in modulating the ER and mitochondrial crosstalk right now. So we're looking at calcium and stuff like that. So yeah, so watch this space, because hopefully we'll be submitting that soon. And then I just want to thank our lab. It's been really great. It's nice having a lab. It's fun. You get to go in every day and do all these cool experiments and talk to people. And then all of our collaborators that really helped with pushing this story out.
Why did you focus on MSS51? Middle of everything. When you developed the rationale for what it did, but what led you to focus on that?
To begin with, pretty much it was just combing the literature. Out of all of these genes, MSS51 was the one that was mainly localized to the same fiber types where we saw our phenotype, and it was doing the same thing that S1P, our Ouroboros data, was suggesting was happening. So that's kind of what led us to, I mean, it was a guess, it was a hypothesis, right? And it just seemed to work out. We're not saying that MSS51 is the only way by which S1P can control metabolism. but we're thinking it's definitely one way by which it's most likely controlling mitochondrial metabolism. The ER mitochondrial story that I kind of like touched on a little bit at the end, it's over. Is it this one? I don't remember. It's one of the red dots, but it's increased. Its expression is increased. And so we wonder if these two are kind of getting modulated by S1P in order to regulate metabolism. And also there are other genes too. Maybe it's, like I said, S1P regulates SREBPs, which is, which controls so many genes, not just lipid and sterile biosynthesis genes, it's controlling ATF6. So we wonder if it's just another arm on the octopus, right, for controlling mitochondrial metabolism through modulating transcription of not just MSS51, but other genes.
I think this is a really amazing example for me to watch. You started with a patient, but you've shown us an example of every way to test your hypothesis, from the RNA to the protein, to the myotubes, to the cells in culture. So I'm gonna thank you for.
Thank you.
That's really what.
Thank you.
Thanks.
Thanks.
We'll
Do you have
So have any GWAS studies been done that have highlighted S1P as being associated with any diseases?
Yeah, so it's been associated with skeletal dysplasia, and then but with regards to muscle, I think there may have been one other study, but it hasn't been published. Like, it was just one other patient that was identified, but still no publication yet on that from the other group. So yes.
So did the patient have less muscle mass, like atrophy, or did she have more muscle mass, or just pain?
I'm trying to remember her chart. We did not see anything with atrophy. It was just pain. Yeah.
I was thinking as you were going through this.
But you would think with the high CK levels, like, and just at like a baseline, something's happening to the muscle, right?
Some sort of term.
Right. We were pretty limited in how much we were allowed to do with the patient, which I totally get. So that's why we switched to the knockout, and why we're making the patient mimic, right? We're making the CRISPR mouse.
And you said this almost happened overnight, which is the other weird thing.
That is a weird thing.
Is what switch was
Right.
thrown.
Yeah. And so it kind of posed a developmental question too. Just to me, I'm not a development dev person, but it just kind of started making me wonder. And so that's why we're also generating, like I said, the PAX7 knockout to see what's happening. I don't know. Yes.
Like any possible treatments for the patient? Or like,
So unfortunately, no. Pretty much, her physicians have pretty much suggested treatments that would help her, function, unfortunately. So yeah. So that's kind of why we're trying to figure out, it's mitochondria. And so we were talking to her physician about that too.
Okay.
Okay.
Now
Thank
we're
you.